Today we will be going over the steps of mitosis and meiosis. However, before we get started, I think it's important to review the anatomy of a chromosome. The entire X-looking structure in orange is the chromosome, while the yellow portion of the chromosome that connects the two legs is the centromere. Now we will split the chromosome in half. Each half of the chromosome is a sister chromatid. They are sister chromatids specifically because they come from the same chromosome. Now, do you notice how the portion of the chromatid above the centromere is shorter than the portion below? This short portion is called the P-arm. Conversely, the long portion is called the Q-arm. Now there's just one more thing we need to talk about before we dive into mitosis: homologous chromosomes. Homologous chromosomes encode the same genes on the same loci, though they can contain different alleles. To signify the difference in alleles, the homologous chromosomes are slightly different colors. Now we can discuss mitosis and meiosis. We will start with meiosis, as it is a simpler process. Mitosis is the process that occurs when a somatic, or self cell, replicates to form two identical daughter cells. The first and longest phase of mitosis is prophase. During prophase, chromosomes are condensed and take on their X shape. Other major markers of this stage include the beginnings of nuclear envelope degradation and the division of centrioles, which will form the spindle used in later phases. The next stage of mitosis is metaphase. During this phase, chromosomes line up at the middle of the cell, and the centromere is connected to cellular spindle fibers. Next is anaphase. During anaphase, the spindle fibers pull on the chromosome in opposite directions. This causes the centromere to break and sister chromatids to be pulled apart and move towards opposite ends of the cell. The final two phases of mitosis are telophase and cytokinesis. These phases tend to blur together, so we will discuss them at the same time. During these phases, the cell divides into two daughter cells and the nuclear envelope reforms. As I mentioned before, these two cells are identical to the cell from which they originated. Meiosis has only one purpose: to create gametes or sex cells. These include things like eggs in females and sperm in males. Unlike the cells in mitosis, these daughter cells are not identical to their parents. Instead, they are haploid and only contain half of the genetic material of the original cell. In order to become haploid, meiotic cells must undergo each phase twice. During prophase 1, we once again see the chromosome take on the X formation, the nuclear envelope degrade, and the centrioles separate. In metaphase 1, homologous chromosomes line up in the middle of the cell and are connected to different spindle fibers. Now comes anaphase 1. During this phase, homologous chromosomes are pulled towards opposite sides of the cell. Notice how this differs from mitosis. Instead of separating sister chromatids, we are separating homologs. Next is, you guessed it, telophase 1. This process still involves the formation of two new daughter cells and the reformation of the nuclear envelope. However, we are not done. This process must repeat all over again in order to create haploid gametes. Prophase 2 is like prophase 1, except each cell only has 2 chromosomes instead of 4. Just like in the other metaphase stages we've seen, in metaphase 2, the chromosomes will align in the center of the cell. In anaphase 2, just like in mitosis, sister chromatids separate as the chromosomes are pulled apart. Finally, during telophase 2, both cells divide in two, creating a total of 4 daughter cells, each with half of the genetic material of the parent.